Today, our topic of discussion will be posterior pituitary. So, if we consider pituitary gland also known as the master gland, it has all the necessary hormones which almost controls all of the other glands of our body. So, as a gland, pituitary can be subdivided into it is a blade-like thing situated at the sella turcica of our skull word, and it has got three divisions. So pituitary, if we draw the pituitary gland, it will look like there will be one stalk, a blade-like thing, and this stalk is attached with the hypothalamus. So this will be the hypothalamus. This is the pituitary stalk, and this is the gland proper. Now just above this pituitary gland, there is near to this pituitary gland, there is the location of the chiasma, optic chiasma. Now, anatomically speaking, if we consider this pituitary gland can be subdivided into, according to most of the author, in two parts. This anterior part, this is known as pituitary, it is located just below the hypothalamus. It is also known as hypophysis. So this part, it is known as adenohypophysis. Because this anterior pituitary, it is concerned with production of some very important necessary hormones. This posterior part, this is a little different. This posterior part, it is known as neurohypophysis. This adenohypophysis, it has got cells which can by themselves produce hormones. So when there are some controlling hormones secreted by this hypothalamus, that will reach via hypothalamo-pituitary circulation, that will reach from hypothalamus to the pituitary. So pituitary, anterior pituitary will produce some hormone by themselves. But when the signal reaches the whole pituitary, posterior pituitary itself, they cannot produce any kind of hormone. So they are not the production side, they are only the release side. So all the posterior pituitary hormone, they are only the release side. And embryologically speaking, this anterior pituitary, it is a gland itself proper, but this posterior pituitary, it is nothing but a continuation of the hypothalamus, which is a part of the brain. And third part, it is particularly prominent in case of lower primates and lower animals. That is this third part, this intermediate part. This is also a division, but they produce some hormones like melanocyte stimulating hormone, something like that. But this part, it is not very prominent, not very active in case of human beings. So anterior pituitary, they usually produce some hormones, very important, namely all of you have heard the name, that is growth hormone. They also produce prolactin. They also produce a lot of tropic hormones like gonadotropic hormones. They produce adrenocorticotropic hormones. They produce thyroid stimulating hormone, etc. Now, this posterior pituitary, this is the neurohypophysis. They, as I have told, they do not produce any kind of hormone. They only secrete it. So how does they do it? If we consider this part, this neurohypophysis, it is made up of a kind of glial cells which known as pituitary. So they are made up of glial cell. So glial cell, that is known as pituitary. These pituitary or these glial cells, their main function is to nourish, is to protect those ascending nerve fibers as well as ascending terminals of the of the hypothalamic neurons. So there are two specifically important hypothalamic neurons. One is known as supraoptic nucleus and another known as paraventricular nucleus. So these two nucleuses, supraoptic, so this is the optic chiasma, one nucleus that is situated, okay, let's go with the blue. One nucleus that is situated here, that is known as the supraoptic nucleus, and another nucleus situated here, that is known as paraventricular nucleus. So these two nucleuses, they have got their own cell bodies. So these nucleus cell bodies, they will be producing some hormones, and through this pituitary stalk, their lower end, that will end in here, that will end in the posterior pituitary, as well as supra. This is from supraoptic nucleus and this is from paraventricular nucleus. Right? So cell bodies will be located within the hypothalamus, within the supraoptic nucleus or paraventricular nucleus, but lower end, that will come to the posterior pituitary to release the hormones. And posterior pituitary, if we consider cell wise, they have got these pituicytes. They are nothing but the glial cells which will provide nutrition as well as provide support to this lower end of this neurons whose body is situated at these nucleuses. So when there is excitation of this supraoptic nucleus or the paraventricular nucleus, there will be release of some hormones. So what are the hormones we are concerned with now? From the supraoptic nucleus, there will be release of ADH or anti-diuretic hormone, and from this paraventricular nucleus, there is secretion of oxytocin. So here from oxytocin will release, and here from ADH will release. Now interestingly, how we came to know about it. Experimentally, what we did, we cut a section through this pituitary stalk. It has been found that initially there was drastically reduction in the amount of circulating oxytocin or antidiuretic hormone. But eventually, if the hypothalamus is intact, eventually this hypothalamus, particularly these nucleuses, they will start to produce both this ADH and oxytocin. But this lower part, this posterior pituitary itself, they will never produce any kind of hormone. Now it has been seen that, it is interesting to note here, it has been seen that though we are saying supraoptic nucleus is producing the ADH, paraventricular nucleus is producing the oxytocin, but they are never ever exclusive. So about 5/6 of ADH is produced by the supraoptic nucleus. If we consider the whole body circulating ADH, 5/6 will be produced by the supraoptic nucleus, but 1/6 will be producing by this paraventricular nucleus, and also the vice versa. 5/6 of the oxytocin will be produced by this paraventricular nucleus, but 1/6 of the oxytocin will be produced by the supraoptic nucleus. So according to this, we will further proceed with our discussion. Now if we consider chemically, both these hormones, this ADH and this oxytocin, both of these hormones, they are chemically very similar. So they are actually chemically very similar. They have got this properties in common. They are 9 amino acid polypeptide hormones. Now they are chemically, I am telling that they are chemically similar, but they are not exactly replica of each other. Okay, so what the difference? Oxytocin has got leucine and isoleucine, whereas ADH have got phenylalanine and arginine. So among these 9 amino acids, ADH and oxytocin, they are almost chemically and sequentially they are almost similar. Only 2 are different. That is ADH, it has got phenylalanine and arginine. In case of oxytocin, it is substituted by leucine and isoleucine. And this is why, as they are almost, they are very closely resembling each other. That is why ADH has got weak oxidative property and also vice versa. Oxytocin has got weak diuretic property, antidiuretic property. So this is the chemical difference between these 2 hormones. Now let's consider these hormones one by one. So first we will discuss about the antidiuretic hormone. So what is the mechanism of action? Now antidiuretic hormone, how it works? Antidiuretic hormone, as the name suggests, it is a hormone which will prevent the water loss. So what are the stimulus? Most important stimulus will be increased or reduced osmolarity of the blood. So increased or reduced osmolarity. So increased osmolarity or reduced osmolarity, that means increased osmolarity means the person is dehydrated. So whenever osmolarity increases, our body's target will be to prevent water loss. So in case of increased osmolarity, ADH secretion will increase. In case of decreased osmolarity, ADH secretion will be decreased. So what do we mean by increased? It has been seen 285 millimole per liter. It is the optimal value of minimal ADH secretion. If the osmolarity increase above 285 millimole per liter, there will be significantly increase in ADH secretion, and below 285 millimole per liter, there will be significant reduction in the ADH secretion. Secondly, one important factor, that is blood loss. Blood loss or dehydration or reduced blood pressure. So interestingly, it has been seen if there is 10 to 15% reduction in the total blood volume, ADH secretion significantly increase. So to prevent the further fluid loss, so that this blood volume can be compensated. Also it has been seen in case of reduced blood pressure, ADH secretion increases. It is proposed that whenever there is reduction in the blood volume or reduction in the blood pressure, there is excitation of this arterial stretching. So arterial stretching as well as reduced excitation of the baroreceptors, they will excite those supraoptic nucleuses, and those supraoptic nucleus excitation will lead to release of the more and more amount of ADH from the posterior pituitary. So actually here only, in this case, osmoreceptors are the sensory organ. The osmoreceptors are the sensory receptor, which will stimulate, in contrast, which will stimulate the ADH production from the supraoptic nucleus. And in these 2 of the cases, the baroreceptors are the significant receptor. These baroreceptors in return will stimulate the supraoptic nucleus to produce more and more amount of ADH. Now about the location of these osmoreceptors, it is still now it is not very sure, not very clear. Some ideas says it is probably located within the supraoptic nucleus itself, but almost all of the scientists these days, they are saying it is located within the organum vasculosum of the brain. So organum vasculosum, it is located at the base of this third ventricle. Okay, within the base of this third ventricle, this organum vasculosum, which is located beyond the blood-brain barrier, they directly senses the osmolarity of the blood. So what happens actually if this area, this organum vasculosum, they are exposed to high osmolarity, so they will get dehydrated, fluid will go out of this organum vasculosum cells, so they will excite the supraoptic nucleus to produce more and more amount of ADH. On contrary, on the opposite side, if the osmolarity reduces, these organum vasculosum cells, they will swell up. So that will reduce the activity of this organum vasculosum, so that will in return reduce the activities of the supraoptic nucleus. So there will be decrease in ADH secretion. So increased osmolarity will lead to organum vasculosum to excite, that will lead to excitation of the supraoptic nucleus and more ADH. So what is the actual exactly this ADH hormone works? So when there is increase in osmolarity or increase in any kind of stimulation, there will be increased stimulation of the supraoptic nucleus. So this supraoptic nucleus stimulation, by next 4-5 minutes, what will they do? They will increase the output of ADH from the posterior pituitary. So increased ADH output from posterior pituitary. So this increased ADH, what will they do? They will work by using enzyme-linked receptor, and there will be increase in activity of adenylyl cyclase. So this adenylyl cyclase increase will lead to increase in cyclic AMP level in the cells itself, those target cells, and this increased cyclic AMP will lead to phosphorylation of membranes. Now remember that this ADH, it has got multiple types of receptors. Particularly important for our discussion is the V2 receptor, which is located at the cells of tubules, collecting tubules and collecting duct mainly. So these, without the response of this ADH, collecting tubule or collecting duct has got very little, also almost minimal permeability to water. So what happens if this is a cell of collecting duct? There will be presence of some vesicles which have got a specific type of channel, water channel, that is known as aquaporin channels. So if this is the cell membrane, normally without the presence of ADH, they will be very much impermeable to water, but they have got a unique capability that they have got some fluid filled, some membrane-like structure which have got a lot of aquaporin channels embedded on their cell membrane. So when there is excitation by ADH, increased adenylyl cyclase, increased cyclic AMP, there will be phosphorylation of this membrane. So what will happen next? This membrane, it will get incorporated within the structure of this collecting duct cell membranes. So there will be appearance of a lot of aquaporin channels. So you can see by this mechanism, by inserting of these aquaporin channels within the cell membrane, of the collecting duct and collecting tubule, these cells will be very much permeable to water. So this is the mechanism of action by which ADH increases the water permeability. So when there is increase in water permeability, water will be absorbed, so increased urine will be reduced more and more in the amount of the fluid, and there will be increase in the osmolarity of the urine more and more according to the osmolarity of the blood. So that optimal osmolarity, that 285 millimole per liter, can be achieved back. So this is the mechanism of action of ADH. Now, what will be happening if there is inappropriately high amount of ADH secretion? So if there is inappropriately high amount of ADH secretion, what will happen? The urine osmolarity, urine electrolyte loss will be very, very high, and body will start to absorb, body will start to preserve water a lot. So there will be edema, there might be some respiratory distress and all these things. So this situation, this symptom, it is also known as syndrome of inappropriate ADH secretion. SIADH. So SIADH, it is a phenomenon where there is inappropriately high amount of secretion of ADH. In these patients, you will find that their blood osmolarity will be very, very low. Their urinary excretion of sodium, particularly sodium, will be very, very high. So these patients will present with hypovolemia, as well as they will present with hyponatremia. So about the treatment of these patients, you have to give some systems which will increase the sensitivity of the V2 receptor, that is known as tolvaptan. So this Vaptan group of drugs are used in case of SIADH. SIADH, you just remember, lot of fluid loss, hyponatremia, a lot of fluid conservation, hyponatremia, along with very, very low osmolarity. Another thing very much common to this SIADH is the cerebral salt wasting. So this cerebral salt wasting, they are almost like a spectrum of SIADH. All of you know that hypothalamus produces this ADH. So if there is some kind of cerebral injury like stroke, there will be very, very much increased amount of SIADH secretion. So again the picture will be same. Hyponatremia will be there. There will be fluid overload by fluid preservation, but interestingly, SIADH requires treatment, but cerebral salt wasting does not require any kind of treatment. If the electrolyte disbalance can be managed for a few days, say 5-7 days, after that very time, if it is cerebral salt wasting syndrome, it will subside by itself because brain will heat itself. Okay, another phenomenon is absence of ADH or very less activity of ADH, that is known as diabetes insipidus. So diabetes insipidus, that means there is diabetes, there will be polyuria and polydipsia, that means subject will drink a lot, subject will urinate a lot, but there will not be polyphagia because they are not glucose intolerant. So in these patients, you will find, if not treated properly, you will find polydipsia and polyuria. Along with that, hyponatremia. So this diabetes insipidus, they can be subclassified into two types. Subclassification are number one, that is central, and number two, nephrogenic. So above it has been seen, if those supraoptic nucleuses, they are affected by any kind of insult, 30% of them due to head injury, 30% of them due to any kind of malignancy or metastasis. Others, there is a very significant amount of other disorders like if there is any kind of pathology in the pituitary, posterior pituitary release hampered, that might occur, or idiopathically there might be reduced production of ADH. So that will lead to central type of diabetes insipidus, and if the kidney itself is unresponsive to the ADH, that will give rise to the nephrogenic diabetes insipidus. So nephrogenic diabetes insipidus, that also can be of two types. First of all, nephrogenic, in case of nephrogenic diabetes insipidus, there might be disturbing the receptors, so V2 receptor defect might be there. So the cells will be unresponsive to adequate amount of ADH, or another term, another thing might occur, that is defect in the aquaporin channel itself. So if there is defect in the V2 receptor, or if there is defect in the aquaporin channel, there might be diabetes insipidus. Now V2 receptor defect, it is both of them are chromosomal defect. This is V2 receptor defect is a X-linked recessive character, and this is one autosomal dominant character. So you can see the treatment, diabetes insipidus treatment mainly combined with either substituting the ADH, if the central cause is there, that ADH secretion is not adequate, you can substitute the ADH, or you can substitute synthetic vasopressin. But if it is a nephrogenic diabetes insipidus, as the V2 receptor aquaporin 2 channels, they are problematic, you cannot compensate it with adding extra ADH. So in this nephrogenic case, main treatment protocol will be giving the subject adequate amount of dehydration, that is adequate amount of fluid, along with that salt substitution if there is severe hyponatremia. But in case of central diabetes insipidus, treatment modality will be giving the subject vasopressin either by injectable route or by intranasal route. So this ends our discussion about the ADH. Now let's discuss about the discuss about the agent hormone oxytocin. Oxytocin, as the name suggests, it is a hormone which is responsible for oxytocins. So that means it is responsible for contraction of the uterus. So experimentally it has been seen, if the posterior pituitary, if the oxytocin hormone production is stopped during labor, it has been seen the labor is prolonged. It has been seen if the oxytocin production is hampered, there will be very less amount of milk letting out, so milk ejection. So by this experimental proofs, we have decided that possibly oxytocin is responsible for mainly two functions, that is number one, parturition, number two, very important function, that is milk letting down. So what are the functions? That is number one, parturition, and number two, milk let down. So these two functions, particularly milk let down, it has been examined, it has been experimented very properly, but how this parturition, how oxytocin works in parturition, it is not very clear to us till now. But let's discuss about the mechanism of action, so it will be easier for us to understand how this parturition and milk let down thing operate. Now again I will be referring to our previous discussion where we have discussed about how these hormones works. So oxytocin usually uses the phosphorylase C pathway. So when there is, in case of parturition, there will be stimulation of uterus, particularly the cervix, and in case of milk let down, lactation, there will be stimulation of areola, there is stimulation of breast plus psychological stimulation. So all these two stimulations, psychological stimulation like seeing the baby hungry or crying the baby, seeing the crying baby, or stimulation by baby suckling, what will they do? There will be sensory nerve stimulation for both of these two things. There will be sensory nerve stimulation. So this sensory nerve stimulation will lead to excitation of this paraventricular nucleus of the hypothalamus. This paraventricular nucleus of the hypothalamus stimulation will lead to increase in oxytocin level. So how this oxytocin works? As I have already told, oxytocin uses the phosphorylase C pathway. So what happens then? Increase in oxytocin, that will reach their target tissue organ. So increase in oxytocin, their target tissue organ in case of parturition will be the uterine musculature, particularly the upper part of the uterus, and in case of milking, milk let down, lactation, it will be the myoepithelial cells which surrounds the ductules or those milk producing glands. So what will happen? When there is high increased amount of oxytocin there will be excitation of a G protein coupled receptor pathway. So increased oxytocin, that will be leading to increased formation of phosphorylase C. This phosphorylase C, what they will do? They will phosphorylate. They will break down the phospholipid component of the cell membrane. So phospholipid component of the cell membrane breakdown will lead to formation of two distinct things. That is, one is inositol triphosphate, and another thing, there will be formation of diacyl glycerol. So this inositol triphosphate, there will be increase. This inositol triphosphate, there will be increase in calcium level. Again, another second messenger, increased calcium level will work through the calcium calmodulin complex. So ultimate result, there will be smooth muscle stimulation. And smooth muscle, it is situated, it is there, smooth muscle is there in case of uterus, that uterine musculature or the myometrium, it is made up of smooth muscle, so there will be excitation by this calcium calmodulin complex. Also myoepithelial cells, they have got their own smooth muscle component, so that will also excite. Now this DAG pathway, what they will do? They will work in two ways. There will be excitation of MAP kinase, that is mitogen activated protein kinase. Okay, so here also there is activation of a kinase, that is MLCK, myosin light chain kinase. So here will be activation of the myosin light chain kinase, which will lead to smooth muscle stimulation. Here in case of DAG pathway, there will be excitation of mitogen activated protein kinase, and another pathway, there will be activation of cyclooxygenase pathway. Cyclooxygenase pathway will excite the production of prostaglandin A2. So this prostaglandin A2 formation, it will directly stimulate the smooth muscle. Also myosin light chain kinase, they will produce a lot of other enzymes. They will directly activate, they will directly excite the smooth muscle component of both the myometrial smooth muscle as well as those of the myoepithelial smooth muscle of the breast. So what will happen? Final result, the smooth muscle stimulation, there will be increased uterine tone. So there will be parturition, that is expelling of the baby out of the uterus. So final result will be, and second thing, myoepithelial cell excitement. So that will lead to milk ejection. So interesting thing to note here for you, that is milk production. All of you know milk production is done by always prolactin, but that milk cannot get out in the ductules so that baby can suck out without the presence of this oxytocin. So when there is presence of oxytocin, initially there will not be any presence of oxytocin, but seeing the baby crying, but when the baby starts to suckle, there will be excitation of this paraventricular nucleus and there will be oxytocin secretion. So initially, for initial few seconds, the baby will not be able to suck anything out. So initial suckling will be a dry suckling. But when proper stimulation has occurred, when the oxytocin has started to work properly, there is formation of myosin light chain kinase, there is formation of MAP kinase and prostaglandin E2s, there will be ejection of the milk. So initial few suckling will be dry suckling, then after that milk letdown or milk ejection will start. So that is why for a successful breastfeeding, these two pathways, both the prolactin pathways as well as oxytocin pathways, required to be intact. Now what are the clinical application of the oxytocin? As I have already told, oxytocin, it is a oxytotic substance that it increases the uterine tone, particularly upper part of the uterus. So oxytocin therapeutically has been used to induce labor. So oxytocin as a drug, it is a very potent labor inducer. It also induces abortion. And before the synthetic oxytocin was available, prior to that synthetic oxytocin, we used to have the knowledge about the oxytocin. So that is why a particular technique that is used by the obstetricians, what they do, they usually during a parvaginal examination, they usually stimulate the cervix by stripping the amniotic membrane from the cervix. So that stripping, that will lead to increase in the sensory stimulus to the paraventricular nucleus, so there will be increased oxytocin secretion. So these two hormones, antidiuretic hormone of the vasopressin and oxytocin, these are the hormones produced by the posterior pituitary. We have discussed both of them. We have discussed their theoretical approach as well as their applications, how the disease involves these glands. So this ends our discussion today.